discussing on how her work can contribute in solving the global pandemic. As we all know, kilala din siya sa tawag na Batwoman because of her expertise in discovering or finding diseases that are bats-related. So ayun, because of her studies, mas naintindihan natin kung ano ba talaga yung COVID. Kaya nga rin tinawag na SARS-CoV-2 ang COVID-19 dahil na-discovered niya na 96% accurate yung genetic component ng COVID-19 sa SARS na nangyari way back. After reading her case studies for the past 16 years, I've read some articles kung saan in-interview siya doon. Ang sabi niya, there are estimated 5,000 undiscovered coronavirus. So, dun pa lang sa statement niya na yun, makikita na natin kung gaano ka-vital yung mga discoveries niya for our current situation and for the future as well. Shi Zengli and her team had discovered how the virus entered their human lungs, and currently they are now working to create a drug that can block it, and hopefully soon they can make a vaccine already. In conclusions, I believe her work can solve the global pandemic that is currently happening right now, and her discoveries can prevent the future pandemics.
These works may contribute in solving the global pandemic because it gives information we need to know about the origin of the coronavirus, like how it started, where it originated, the symptoms of the coronavirus, and how serious it is, the genetic component or sequence of the virus that can help us find or make cure. According to Zengli, coronaviruses have caused two large-scale pandemics in the past 2 decades, which was SARS-CoV and the Middle East Respiratory Syndrome or MERS-CoV. It has been generally thought that SARS-CoV, which is found mainly in bats, could cause a future disease outbreak, which was the COVID-19 or coronavirus 2019. It was called COVID-19 because it occurred in 2019 and the virus started in Wuhan, China. Coronavirus has caused an epidemic disease outbreak in Wuhan, China. The typical clinical symptoms of these patients are fever, dry cough, breathing difficulties, headache, and pneumonia. Disease onset may result in progressive respiratory failure owing to alveolar damage and even death. The disease was determined to be caused by virus-induced pneumonia by clinicians according to clinical symptoms and other criteria including rise in body temperature, decreases in the number of lymphocytes, and white blood cells, although the white blood cells are sometimes normal. New pulmonary infiltrates on chest radiography and no obvious improvement after treatment with antibiotics for 3 days. It also appears that most of the early cases of people with pneumonia disease had a contact history with the original seafood market where the virus had started. However, the disease has progressed to be transmitted by human to human. Samples from several patients with severe pneumonia investigated by the laboratory investigating coronavirus. They used first-pan coronavirus PCR panels to test the samples, given that the outbreak occurred in winter season and in the market, the same environment where the SARS infection started. They found five samples to be PCR positive for coronavirus. One sample collected from the bronchoalveolar lavage fluid was analyzed by metagenomics analysis using next-generation sequencing to identify potential etiological agents. Of the 10,038,750 total reads, of which 1,582 total reads were retained, after filtering of reads from the human genome, 87.1% sequences match the sequence of SARS-CoV. The virus genome consists of 6 major open-reading frames that are common to coronaviruses and the number of accessory genes. Further analysis indicates that some of the 2019 nCoV genes shared less than 80% domains in ORF1 lab that were used for coronavirus species classification were 94.4% identical between 2019 nCoV and SARS-CoV, suggesting that the two viruses belong to the same species. SARS-CoV Then they found that a short region of RNA-dependent RNA polymerase from a bat coronavirus, bat-cov rat-G13, which was previously detected in Rhinolophus affinis from Yunnan province, showed high sequence identity to 2019 nCoV. They carried out full-length sequencing on the RNA sample. Simple analysis showed that 2019 nCoV was highly similar throughout the genome to rat-G13 with an overall genome sequence identity 96.2% using the aligned genome sequences of 2019 nCoV, SARS-CoV, and rat-G13 and previously reported bat SARS-CoVs. No evidence or recombination events was detected in the genome of 2019 nCoV. Phylogenetic analysis of the full-length genome and the sequences of the RD, RP, and spike showed that for all sequences, rat-G13 is the closest relative of 2019 nCoV and they form a distinct lineage from other SARS-CoVs. The receptor binding spike protein encoded by the S gene was highly divergent from other coronavirus with less than 75% nucleotide sequence identity to all previously described SARS-CoVs, except for a 93.1% nucleotide identity to rat-G13. The S genes of 2019 nCoV and the rat-G13 are longer than other SARS-CoVs. The major differences in the sequence of the S gene of 2019 nCoV are the three short insertions in the N-terminal domain as well as changes in 4 out of 5 of the residues in the receptor binding motif compared with the sequence of SARS-CoV. Whether the insertions in the N-terminal domain of the S protein of 2019 nCoV confers salicylic acid binding activity as it does in the MERS-CoV needs to be further studied. The close phylogenetic relationship to rat-G13 provides evidence that 2019 nCoV may have originated in bats. Zengli rapidly developed a qPCR-based detection method on the basis of the sequence of the receptor binding domain of the S gene, which was the most variable region of the genome. The data showed that the primers could differentiate 2019 nCoV from all other human coronaviruses, including bat SARS-CoV, which shares 95% identity with SARS-CoV.
So I'm going to talk about case studies conducted by Shi Zengli and her fellow researchers that happened way back 2003, which is the SARS Severe Acute Respiratory Syndrome. So the pandemic started in November 2002 and was brought under control in July 2003. Accumulated studies have several findings that indicated an animal origin on the basis of the following findings. Number 1, studies indicated that SARS-CoV is a new virus with no genetic relatedness to any known human coronavirus. Second, studies found no evidence of seroprevalence to SARS-CoV or related viruses in the human population. Third, serologic surveys among the market traders during 2002 to 2003 outbreaks showed that antibodies against SARS-CoV were present at a higher ratio in animal traders than control population. Next is SARS-CoV isolated from animals in market were almost identical to human isolates. Next, molecular epidemiologic analyzes that human SARS-CoV could be divided into 3 groups, early, middle, and late phase, and the early phase isolates were more likely related to animal isolates. Human SARS-CoV isolates from 2003 to 2004 had higher sequence identity to animal isolates than human isolates from 2002 to 2003. So there are certain evidence to prove that SARS-CoV came from animal. 25 animals are sampled, which is yung virus is na-detect in 3 mass found civets and 1 raccoon dog. Currently, greater than 10 mammalian species have been proved to be related to this virus. Rats were also potentially susceptible animal that may have a role in transmission and spreading this SARS-CoV. The first human with SARS have no contact with any animals except rats. So, bats have been identified as a natural reservoir for an increasing number of emerging zoonotic viruses, including henipaviruses and variants of rabies viruses. Several horseshoe bat species have a close genetic relationship with the coronavirus, associated with severe acute respiratory syndrome, or also known as SARS. The discovery of bat SARS-like coronavirus and the great genetic diversity of coronavirus in bats. Sequence analyses indicated the existence of a much greater genetic diversity of SARS live in bats than of SARS-CoV civets or humans, which supports that bats are natural reservoir for it.
So we're going to talk about one of the case studies that Shi Zengli conducted together with his team way back 2006, which is the bats and the virus. And we all know that the bat is the only flying mammal and count more than 20% of the extant mammals on Earth. Were recently identified as a natural reservoir of emerging and re-emerging infectious pathogens. And yan, so yung pathogens is virus or microorganisms that can cause disease. So re-emerging and re-emerging infectious pathogens kasi syempre yung before kasi is SARS, and then ngayon parang nag-re-emerge sila ulit, naging COVID or the coronavirus disease. And then, many studies focus on bat viruses that cause severe domestic and human disease. And then, however, many viruses were found in apparently healthy bats, suggesting that bats may have a specific immune system or antiviral activity against virus infections. Yun, so, may mga healthy bats na madaming viruses sa katawan, and siguro meron silang antiviral or kung ano mang protection sa katawan nila para hindi sila tamaan ng infection or hindi sila ma-infect dun sa viruses na nasa katawan nila. Therefore, basic researchers for bat immunology and virus host interactions are important for understanding bat-derived infectious diseases. Yan, so yung ano kasi zoonotic viruses is when it comes to carrying viruses that can be transferred to other species including human, that is so called zoonotic viruses. And then these flying mammals host over 60 zoonotic viruses. This is rivaled only by rodents that carry a wide range of bacteria, viruses, protozoa, and helminths. Yun, so, alam naman natin na yung bats is nagko-contain ng madaming viruses na pwedeng makahawa, especially sa ating mga tao, kapag kinain natin. Ayun nga, so, guys, hindi talaga healthy yung kumain ng bats. Although ginagawa silang pulutan, ginagawang considered as exotic food, especially dun sa public market ng Wuhan, China. And we all know naman na hindi naman talaga galing sa public market ng Wuhan yon. Something like parang accidentally nag-leak siya dun sa from Wuhan lab, pero parang dinedeny siya ng China. Ayun. And then bats also carry more human pathogens than other animals because bats prefer to live close to one another, like humans, spreading respiratory viruses like the flu during winter, giving plenty of opportunities for pathogens to spread between the bats. Yan, so tayong mga tao kasi, minsan kasi kaugalian na natin na kunwari mag-sneeze tayo, hindi tayo magko-cover. Syempre yung virus natin nag-i-spread out, mahahawaan yung mga taong nasa paligid natin. Kaya, syempre, kailangan talaga nating mag-ingat and well-sanitize dapat ang mga kamay natin, at kapag madumi ang kamay natin, hindi tayo hawak sa mata, sa mukha, sa bibig, sa ilong, kasi sa parts ng mukha nang madumi yung kamay natin. Kasi viruses can spread all over and pwede tayong makahawa ng makahawa.
Who is Shi Zengli? What is her relevance to this global pandemic, and what is her connection to P401 facility, and how can be she possible? Paano may posibilidad na siya din makagamot nito? So, first of all, basic information. Shi Zengli is a 35 years old Chinese virologist and a writer. She is also a researcher from Wuhan Institute of Virology, and together with Shi Zengli, they're the ones who discovered that SARS is from bats. Diba SARS kasi, diba? Pare-pares kasi yung SARS, MERS-CoV, and COVID-19, lahat sila galing doon sa virus na tinatawag na SARS-CoV-2. And it is all from bats. Diba? We all know naman, diba, na ang origin ng virus ito is solely from bats. So, paano nga ba nila nasabi na galing sa bats yun? So, anyways, yung sinabi nila na the virus originated from bats, diba, nagsimula nga sa SARS yun, they stated it in their case study from 2005. And yun. So, let's go back to the question na paano nga ba nila nasabi na galing sa bats? So, first is, bats are already known as a main source of emerging and re-emerging infectious pathogens. So, she can be the key to this global pandemic kasi siya nga, di ba? Siya nga yun. Tsaka yung team niya, yung nakadiscover ng virus dun sa Wuhan lab. And so, that means, kung sila yung nakadiscover, sila yung familiar dun sa genetic components ng virus na yun. So, in other words, sila lang din yung may familiarity at may advantage na makadiscover kung ano ba yung pwede maging gamot doon. Kasi nga, sila lang yung familiar dun sa genetic components ng virus na yun. Then, afterwards, lumabas na yung mga claims and conspiracy theories na galing mismo doon sa Wuhan lab yung coronavirus na sasabi nila. Kasi, may nagkaroon ng konting possibility na totoo yun. Kasi, according to some articles and research, yung kiniklaim ng team ni Zengli na bot is hindi present doon sa market. Kasi sabi nila galing doon sa kinakain ng tao. Eh kaso, yung kinakain ng tao na yun, binibenta yun sa market kung saan nagbibenta ng mga exotic animals. Kaso, yung bots na pinagbasihan ng coronavirus is hindi siya present dun sa tindahan ng mga exotic animals. So, remember, way back 2003 during SARS outbreak, they stated that the origin of the virus is from the bats. Again, bats kasi are really known to have an increasing number of emerging zoonotic viruses and different variants of rabies viruses. So, basically, it is not freaking safe to eat bats. It is not for us to eat. Pero, we're not talking about naman that. I just have to banggitin lahat yung kung anong pinagmulan ng lahat na to. Kasi nga, ano nga ba ang relevance ni Zengli dito? Kasi nga, sa kanya nagsimula lahat ng study na to, lahat ng claims na to. Ayan yun. Kanina nga, nabanggit ko na wala sa market yung sinasabing bats na nakitaan ng team ni Zengli ng virus. There are different type of groups of bats kasi talaga. So, pwedeng yung nasa sa market is ibang uri ng paniki. At yung nasabing pinanggalingan ng virus ay pwedeng ibang uri din ng paniki. At yung specific type, yung sa pinangkitaan ng virus is, yung specific type na yun ay tinatawag na genus Rhinolophus. Sir, sorry for the old unwanted noise. So, ayun sir, sorry sa mga maririnig mo nag-iiyakan dito. I can't do anything about it. They're kids. Wala nga naman patigilin ko silang umiyak. So, ayun. Tuloy na nga natin. Kasi yung binanggit kong species na ngayon, na yun is known as the reservoir host ng malaking bilang ng mga viruses na sinasabi na merong genetic relationship with the virus that is associated with SARS. This is according from one of Zengli's team case study titled Emerging Infectious Disease from 2006. And that said, so her team again claimed that the novel coronavirus is from bats as they studied it on their laboratory. So one of Zengli's and her team case study from 2010 titled Infectious Genetics and Evolution talks about the hantavirus outbreak that are associated with lab rats in Yunnan, China. So, bakit ko nabanggit yung about sa hanta outbreak? Kasi just like MERS-CoV, SARS-CoV, and nCoV outbreak, the reason behind the human infection is because of the zoonotic transmission of the virus from the Wuhan lab in China. So, that supports the claim of the majority na galing mismo sa Wuhan lab yung nCoV. Pero kung totoo yun, we don't know if it's intentionally or not. But I'm not here for that. I am here to discuss about what is her relevance, her possible contribution, and some case studies of her team that might answer our questions. So, what is her connection to the P4 Wuhan facility? Basically, she is the second lead in command in P4 Wuhan facility. So, what is P4 Wuhan facility? P4 Wuhan facility is one of the top Wuhan laboratory. So, summary is, kaya siya relevant kasi una, sila yung nakadiscover kung anong pinanggalingan nung mga outbreak na yon. Pangalawa, bakit siya may posibilidad na makatulong sa pag-solve ng pandemic nito kasi nga siya lang at ang team niya ang pinaka-familiar about dun sa genetic components ng virus na yun. And bakit tayo naghanap ng case studies niya? Kasi we have to know kung ano ba yung mga pinag-aralan nila from the past, bakit nila nasabi, kung ano ba yung basis nila, bakit nasabi nila na galing sa bats, and that can na pwedeng maging patunay na hindi talaga man-made yung virus.